on. Hello, everyone, and welcome to another episode on the Biofrontier Files. I think this is episode 13, and today we're joined by Dr. Insco.
Hello, everyone. Aditya and Dr. Insco. Why don't you go ahead and just introduce yourself a little bit for our viewers?
Yeah, so my name is Megan Insco, and I am a principal investigator of a lab that I started about 2.5 years ago at the Farber Cancer Institute. And I also see melanoma patients as an MD, and I specifically a medical oncologist, half day a week. And so that's my current job. I also have two wonderful kids, one 8-year-old and a 10-year-old, and I love to play ultimate frisbee. Frisbee, excuse me.
Sure. So let's get started. The first question is, how did you really get introduced and interested into science when you were growing up?
Yeah, so I think it was always a natural fit for me to get involved in science. I think from a very early age, I was always really curious, and my dad minored in physics at like a small community college, but he always had a fascination with that. So he would get Scientific American at home, and he would try to teach me about the articles he would read. So I have memories of doing that as a small kid. We made a tic-tac-toe like computer one time where we basically played out like every possible move in tic-tac-toe and like the order of which you had to do stuff, and then like coded this into like a system that you could read. So that was always kind of a natural fit for me. But then what really focused my efforts was when I was in middle school. My mom got breast cancer, and then my mom got really sick, and then she died when I was in early high school. And so that really focused me. Like I already like science, but then when that happened, I was like, whatever I do, I want to help cancer patients. I didn't know at that stage whether that was going to be through research or medical care or fundraising or just, supporting people, but I knew that was going to be a major part of what I did. So kind of, yeah, definitely focused me. And then in college, I still wasn't quite sure, but I just had all the pre-med stuff. I actually pre-meds are not my scene, so that was really hard for me. But during the actual pre-med activities, I found myself just naturally able to speak to patients in a way a lot of my colleagues or my people I was in college with weren't able to. It was just felt right, so I kept pursuing that. But I didn't really make that decision until my senior year of college. So I knew I needed to have a job. So I applied for research tech positions and got a job at WashU in St. Louis and had an amazing boss. And she taught me I already knew I liked science, but she taught me how to think like a scientist, uh, like necessary and sufficient and present and just the logic. And it was a beautiful system we worked in. And I was really taken with how she was a young PI too. I was just amazed at what she did. So I applied to medical school, went to medical school, and then applied for PhD programs in medical school. So this is a long story, but that's kind of how I got to where I am now.
Yeah, so now that you are where you are as both MD and a
Mhm.
PhD doing research and on the clinical side as well, what do you really find most rewarding about your job?
What I love about my job is that it is so varied. Like it changes on a daily, sometimes hourly basis. Like I'm definitely never bored. Um, so what I find rewarding, similar to that, it depends totally on the day and the time. And I think for some things, obviously the patient care can be extraordinarily rewarding, can be also extraordinarily challenging, like interpersonally, but also, just extremely sad at times when things don't work. But it can be really rewarding when you really help someone get better, or which we actually can do in melanoma these days. Or it can also be super rewarding when you help someone die well, which may sound crazy, but there's bad deaths and there's good deaths, just like there are bad and good lives. Like the everyone's going to die. Like the quality of that and how we plan it out, and meeting people's visions and goals about that. So I feel like really satisfied when I've helped someone uh, live their life out the way they want it. And then from a research standpoint, there are those rare moments where you kind of put together that something is really important. And those are also extremely satisfying. I guess that's more from the research content, kind of like those aha moments people talk about. And then I think the last thing I find extremely satisfying is when I teach somebody to do something and they do it really well. That's extremely satisfying, especially if they could do it better than I did it myself. Then I just know that I really that I did a good job and that I'm like giving back, or that's, helping the world out. So
Absolutely. So the next question is, what is your current research really about? For our viewers, you work with RNA and melanoma and for finding your drug. So just kind of, get into that a little bit because our viewers would definitely want to know.
great. So we are really interested in finding new therapeutic targets for melanoma. Melanoma, it's a cancer of pigment-producing melanocytes in the skin, and it can be really deadly when it metastasizes or is not treatable by surgery alone. And that's really despite some major breakthroughs in therapy that have occurred in the last 10 years. So I'd say 50%, maybe 60% of our patients now do really well in existing therapies, but there's still like tens of thousands of people who are dying of this disease like in the last couple of years. And so we can do better. And I think we need to think about it creatively and take a step back and find other pathways that really work. So what we have right now is we have immune therapy agents, which help the patient's own immune system to activate their immune system to attack the tumor. And we have some kinase inhibitor therapies against what's called the MAPK pathway. It's just a specific signaling pathway in cells that work really well in certain contexts with patients that have certain mutations. But I think we need like a third or maybe a fourth major target to go after. And so what I got interested in during my postdoctoral science training is I found a new tumor suppressor, and it worked in a really surprising way. So this gene was called CDK13. It's mutated in about 4% of melanoma patients, so not a huge number, but what was really cool is that it normally causes the cell to clean up mistake RNAs, take the trash out, so to speak. And when it's mutated, these trash RNAs like build up, and they actually are sufficient to cause worse melanoma. And this is really interesting because it's a whole new way of thinking about it, a whole new tumor suppressive pathway. And whenever something like that is revealed, there's going to be therapeutic options either through cancer or for other diseases. We just have to understand the molecules, what they look like, how they're interacting with these RNAs, what these RNAs exactly are, to figure out how to control it to help humans. So this is most of what my lab is focused on. A smaller portion of my lab is focused on understanding the drivers of a rare subtype of melanoma that is very lethal called mucosal melanoma. I've taken care of a number of these patients, and they just don't do as well. They don't respond as well to our existing therapies. The genetics of the tumor are entirely different. And so we take human cancer genetics, and then we make those genetic changes and using zebrafish is the model we use. And we try to figure out like what's causative. So those are the two major parts of my lab.
Yeah.
And just
Oh,
okay, sure.
so why really zebrafish models? Why not a different animal or species?
Yeah, so I think there's a couple of reasons there. One is that I have worked with mice before, and frankly, it was a little hard for me to work with mice. They're kind of cuddly, and I know maybe that sounds weird, but I just had a hard time working with them. But if you're going to do cancer research, you really need to work with a model organism that has like vertebra. So like basically something that's more similar to humans than a lot of the other existing model organisms that are out there. Zebrafish is really the next closest thing. And then when I was interviewing for positions, I still interviewed for postdoctoral positions. So when I was training with a number of labs that did mouse genetics, because I was really interested in I think that science in an organism is really important. Like we are complicated. There's a lot of aspects going on. And I think you need a strong phenotype, like meaning a change you can find that you can follow. And a lot of times those are much more clear in model organisms, as opposed to in just some other models like cells growing in a dish. So I really wanted to have a model organism, and you need one with a vertebra, and mice were kind of cute. So I looked for zebrafish labs, and then I trained with Len Zahn, who's at Boston Children's Hospital. He really helped make zebrafish a mainstream adult model organism for cancer. And I'm so glad I did, because I learned a ton. It's a fantastic system. It was actually the first melanoma model was built in zebrafish, not in mice. So, and he was part of that. So it's I guess those are some of the reasons. Some other reasons are that they grow really quickly, and we can put a ton of animals in one tank, and they're happier that way. Actually, they would rather have 30 or 40 animals in a tank. They get stressed out with just one. So from a housing perspective, like, they're happier, and then we're happier too, because we can get more science done in a small amount of space. They're less expensive to take care of, and they grow faster. You can also, they can be see-through. So we have certain genetic changes. So we can actually, for this mucosal project, some of our fish actually will get tumors inside, and we can actually watch them occur in these see-through. We call them Casper fish, like the ghost. Yeah. So it's also, we can do really rapid genetics. I think this is starting to become more true for mice, but for years, mouse genetics was quite slow because of all the crosses you had to do. But we can do a lot of our experiments in the first generation. So basically, we can introduce some genetic changes into the fish when they're one cell stage embryos, and then we can just watch them grow up and observe all the changes we want to. And that's the experiment. And we don't have to keep crossing those and then crossing those and crossing those. We can keep starting from point zero, F0.
So kind of related to what you just talked about, you're talking about how you were talking about the new fast genetics in mice and things like that. So how do you stay updated with these new developments in, not only your field, but the related areas, like either it's melanoma or model organism handling, or it's just a new drug therapy? How do you stay updated?
Yeah, it's a great question. It's a great question, because there's so much happening all the time. I try to, I have a few approaches. So one is I try to go to a couple meetings a year that are really high yield. I think that's where I get exposed to the most. Like, I go to meetings. I'm a young professor right now, like an assistant professor. I'm not one of these fancy full professors, but I try to go to the meetings where those fancy full professor Nobel Prize types are hanging out, and I just listen, and I listen as much as I can. And I get the information firsthand. And from those conversations, I figure out what's set, like, what should I have known? Like, what is like absolutely, everyone, like, I should know. What isn't really clear and they're still debating. Like, that's important too, because I may not influence that debate, or I might if it's my exact field. But if it's not, I'm just not going to touch that. I'm going to let someone else figure it out, and then I might try that experiment. Who do I need? Who to ask about what? So I do a lot of that at meetings, and it's been super helpful at starting collaborations that way, and just asking, like, how do I learn that? I read a lot. I think I read in kind of a unique way, where I don't usually read start to finish, although maybe I should more sometimes. But I'm really driven by the questions I have. I really want to answer from my own science. And then I'm really driven, as my reading is guided, to answer those questions. And so lots of times I'll pull out, and I always try to focus on really high quality papers in science. And that just comes with practice, but I've looked at enough figures and read enough things that I can tell what I trust and what I don't at this point. And once I have those high quality things, then I'm kind of judging. It's kind of like, have you ever tried to put together a recipe and you look at like 10 different recipes for something, and you're like, oh, well, they all use potatoes, so that must be important. But this one cooks for 2 hours, this one cooks for 20 minutes. So like, maybe that has some flexibility, like that kind of thing. So I try to do that with my reading.
Got
And
it.
then in the Boston ecosystem, there's just a ton of, there's basically, you run into people constantly, and I just by happenstance get help and get updated about things I should know.
Yeah, definitely. Moving more on to like with AI and
Mhm.
improved technology.
Mhm.
Since you started and with AI as well, how do you think it's going to impact your field, and how are you integrating it into your everyday lifestyle and work?
Yeah, that's a really great question. There's a lot of discussion about that. I think that I haven't actually integrated it yet. There's a lot of discussion about what we should and how we should be doing this. I think I have one computational biologist in my lab. She definitely uses it for, because you can ask ChatGPT for help with code. And like, I think everyone does that these days. It's like very accepted. So Now, things I can't use it for is I can't use it for writing manuscripts. I can't use it for writing grants. Like that's expected to be me. I can technically use it for writing emails. I have one colleague who uses it to help her write tough emails. So I guess I haven't tried it yet. I think that there's a lot of things that patient care wise, I think it's going to be really helpful for, but I think a lot of it is we know what needs to happen, and maybe AI will help us track some of that, like what happened for a patient. But a lot of it, the patient just has to it has to be communicated to the patient, and the patient has to know to show up and do these things, and then it has to be checked off. So I don't know if AI can help with some of that. One place it is happening in clinical practice, or they're starting to trial it, is they're starting to use AI to monitor our visits to help with documentation and billing. So that's an interesting thing. It's not like prime time, but they're starting to test that. That would be great, because if I didn't have to spend another hour or two at the end of my clinic session writing notes, if it literally just captured what I did, that'd be amazing. But I don't trust the accuracy, but that would be awesome. But in the science stuff, I don't know. I don't think we're there yet. I think AI is really good at taking something you already understand, or somebody really understands, and then distilling it for other people, like language. Like a lot of people really understand language. You put this huge data set in, and then it can speak. But for a lot of the stuff we're dealing with, no one understands it. So what are you going to train it on? And then, yeah.
Got it, got it. So it's like an electronic health record, really, and it's like you're thinking about using. I think you mentioned the language. Someone said something and try and use that. So it seems like it's heading towards a more of a AI direction and more
Yeah.
integration. Yeah.
I think that will
So
happen.
probably will, and most fields too. All the professors we've talked to so far, all of them have said that yes, they've integrated it. It's introduced to their
Mhm.
field, right? So I think now we're gonna ask one more question.
Mhm.
And this question is just what is a typical day for you? I know you mentioned how you like how variable your days are, but
Mhm.
what would be a typical day?
Yeah. So do you want one of my clinic days, or do you want one of my not clinic days?
I
Could we
mean,
get
both would
both?
be good.
Yeah.
Okay, so Mondays I are my clinic day in the morning. So typically then I'm headed in. So I like to go in early on Mondays just to kind of get a start, because so much stuff comes in during clinic. So I try to get a lot of emails and things off my plate. So usually my husband takes the kids to school or camp that day, and then I walk to work, which is lovely. So I have like a 25-minute walk, and I walk in, and then I'm just, the patient stuff is pretty easy to talk about because everyone kind of knows what a patient visit's like. But so I see patients for the morning, sometimes new patients that have longer slots for. Usually it's pretty routine these days. If there's going to be an emergency, I usually know it's coming because the patient has already contacted me. But occasionally something crazy will happen and things get derailed. So, that can, if someone's really sick, or maybe I'm covering for someone else and they have someone who's really sick, or, surprises can come out. So I usually finish up in clinic. By the time I finish my notes, et cetera, I'm usually finishing up around 1 p.m. And then I have a variety of meetings in the afternoon where I check in with my different lab mates, check in with a couple of postdocs, and then I have, I guess, actually, I check in with 3 people depending on what's going on. Like if we have a manuscript that we're writing, I meet with people more frequently because there's always like smaller things. Uh, yeah, it doesn't sound very exciting, but that's like a standard Monday. And then I get a little bit of time for work myself. I maybe get like an hour or so to go through emails, or to do a little bit of writing, or just to triage the rest of my week. Mondays and Tuesdays are my big writing days. So that's I love those days. They're quiet. I get to this today. I spent a bunch of time working on my aims for a big grant that I'm writing, my first R01 grant in the fall. And that is helpful to help dream up experiments. And then, sorry, I guess this isn't like one typical day, but a typical Friday, I meet with everybody in the lab on Friday. So I have like tons of meetings, and I have my clinical meeting in the morning where we run patient cases. So that's where I get a chance to, if I have trouble or I don't know what to do with a patient, or I'm some sort of challenge I'm having, I can present it at that conference and get practical advice. So it's kind of like my home base for my MD side. So I don't know. That wasn't really what you asked for, but Anyway.
No
I mean,
worries, no worries.
that's a busy life and also an interesting one. I mean, you get both angles, I guess, and the political and the research. So
Yeah.
that's always good. So we've started this thing where we ask a final question, like a fun question. So not about your job or anything. What do you like to do for fun?
Yes, I mentioned this earlier, but my side passion is ultimate frisbee. I've been playing since I was in I started in late high school because a couple of my friends went to college and started playing, and then I played in college, and we were terrible, but it was really fun. And then I played on like a club team there, and then I went to California, and during my MD PhD, I played competitively and played on a team that won the national championships for women, and like trained a bunch for it, and then it got too busy, and
Wow.
I had to go back to medical school and like
That's
actually
impressive.
and actually like work really hard hours and things, and so it was not possible to keep that up then. And then I trained in medicine. Actually, I still played on a top in my last year of medical school. I actually helped start a team that didn't practice, and we won nationals for in the co-ed division. So I started that, and then basically I've squeezed it in wherever I can. I just get great joy out of it. I get great joy out of chasing a disc, and I love the endurance aspects of the sport and the explosive aspects of the sport. So I actually organize a team now that's for women over 37. So we're still doing it. It's I've got a
That's
one. We even have a 57-year-old on our team, and she's still going
wow.
strong. Yeah,
That's amazing. What more
yeah,
would you
it's
want?
yeah. So anyway, that's my side.
Got it, got it. Well, like we said, that was the last question. So thank you so much for joining our podcast today. And we really appreciated your very honest and very practical aspect of your life, your career, your research. Everything was very nice. We really appreciate it.
My great pleasure. Thanks for inviting me to do this, and good luck to you both.
Thank you so much. Bye.
Thank you. Bye-bye.